All right, now we get to start talking about DNA. And DNA is known as the molecule of life. It is the recipe, right, for making proteins. We've talked about proteins, that they pretty much do everything within the cell. But how does the cell know how to build those proteins? What is the blueprint for determining the structure and thus the function of those proteins? And that's DNA. So we're going to start talking about DNA. This opens the book to talking about genetics, which I know a lot of you are excited about learning about. So the first thing I want you to do is I want you to think anything you can remember about DNA, either stuff we covered earlier in the year or things you talked about maybe freshman year with Miss Bebeck or whoever you took biology with. Perfect. Awesome. So long ago-ish, and when I say long ago-ish, I don't mean like a really long time ago. I mean within the last 100 years, we had no idea that DNA did anything. In the 1920s, which was 100 years ago, we had no idea that DNA was the genetic material. So people had no clue why children looked like their parents, right? Or why they looked like their siblings. Why did they resemble each other? How do cells reproduce and make copies or copies of themselves? How does all this stuff happen? How does it work? So I need to cut out some of my coughing fits because I still have this upper respiratory thing going on. So what type of molecule is responsible for differences? Now, you may be tempted to say DNA. That is the thing that is the underpinning blueprint for it. But when we talk about what actually physically causes your differences, that's proteins. Proteins, pretty much, as I said, do everything within the cell. What causes skin tone to be different color? The protein melanin. What causes the nose shape that you have? It's due to cartilage and muscles within your face that pull your nose in a specific way. Lactase is an enzyme that breaks down milk. People can be lactose intolerant. Some people are, some people aren't. Does that gene turned on or off in those people? Growth hormone is a protein. How much do you make? How often do you make it? When does it stop being produced to cause you to grow substantially? There's all sorts of things that relate, and it's way more complicated than that. But what causes our differences? Those are proteins, and our bodies make over 30,000 different proteins, and each cell produces different types of proteins, so each cell can do its individual job. So just a quick review of the levels of protein structure. Remember, primary structure of a protein is the order of the amino acids. Remember, those amino acids each have different characteristics based on those R groups that they have. Some R groups are polar, some are non-polar, some are charged, some are form specialized disulfide bridges with other ones. So the order of the amino acids is very important, and that's the primary structure. Secondary structure is how it starts to form into alpha helices and beta sheets, as you can see down there. Remember, that has nothing to do with R group interactions. That's just hydrogen bonding between adjacent amino acids, amino groups, and carboxylic acid groups of adjacent amino acids. Then there's tertiary structure, which is taking one whole subunit of protein, one long chain of protein or a polypeptide, putting it together, and that starts folding based on R group interactions. And then there's quaternary structure. That's when you put multiple subunits together to all give a protein its overall job and its structure and function. So remember, why people are wondering, why do you look like your parents and your siblings? How do organisms pass information to the next generation? And what is the genetic material that is controlling this, that is being passed so that everyone knows how to kind of make the proteins that are necessary for life? How does your cell know how to build each of these little molecular machines that we have been talking about? Receptors, ligands, a lot of them are proteins, signal transduction pathway proteins like kinases, enzymes that produce second messengers, transport proteins, the sodium-potassium pump, co-transport mechanisms, microfilaments within the cell that the diet and kinesin molecules walk on to move vesicles between the endomembrane system. All of these are made of proteins. All of these enzymes, catalase, like that was in the potato slushy that we're doing that lab on, right? So how does your cell know how to build these? So what we know now, this is the general gist, right? So DNA is like a recipe book. It is like the
It is the instructions for building these individual proteins. And the 4 letters of DNA, or the bases, the adenine, thymine, guanine, and cytosine, A, T, G, and C, depending on their order that they're present within the molecule, you can create different words. And those words correspond to specific amino acids. So it allows for the cell, or the ribosome, which is the thing that makes these proteins, assembles proteins, to put these amino acids in the order that is dictated by the DNA. And each of us has our own unique recipe book, though most of our DNA is actually very similar, because on the cellular level, we're very similar. But there are some differences that are found within our DNA that allow us to have the differences that we do, characteristics-wise. Obviously, your experiences in life can affect that too. It's not just the DNA, but DNA is kind of like the hardware. It helps to lay down the hardware that you have, and then the software that you put down in your body is kind of your experiences that you have. So what we're going to focus on in this video is how do we figure all this out? How do we know that DNA is that DNA was the genetic materials? The first experiment that's really important to understand is Griffith's experiment. So this is one of the first times that we've actually gone through some experiments that they expect you to understand on the AP exam. So Griffith's experiment, what he did in 1928, so less than 100 years ago, right? He did an experiment to determine whether a trait could pass from one cell to another, and he used virulent or infectious bacteria and avirulent non-infectious bacteria. And essentially, virulent bacteria formed what's called a biofilm on the outside, and that's the trait that it had that made it more dangerous. The avirulent one did not produce that biofilm that causes it to, or it's a protein coat on the outside that allows it to make a biofilm. So the non-infectious one didn't have it. So he transformed the avirulent or not dangerous one to virulent that could kill mice. Now, before I discuss Griffith's experiment, I want you to explain something to you that bacteria do that's unique, that they didn't really know about then. Bacteria can pick up little pieces of DNA from their environment. So they don't have to sexually reproduce to pass genes from one to another, or even have mutations. They can simply, once one bacteria dies, sometimes its DNA ends up in the environment. Bacteria can pick that up and actually gain the traits of the bacteria that died next to it. So it's important to understand how this actually worked. So what Griffith did, very simple, he took dangerous bacteria, which are the one with the capsules on the outside, that's what causes it to form biofilms, and he injected that into a mouse, and that killed the mouse. And that makes sense, right? Because it's the smooth cells are the dangerous ones, okay? He then took ones that were not dangerous, the rough cells or the avirulent ones, and he injected into a mouse, and the mouse was fine. It survived because it wasn't injected with a dangerous bacteria. That all makes sense. But what was interesting now is this next thing that he did. So then he heat killed. So he killed the dangerous cells, right? And then he injected the dangerous cells into the mice. And the heat killed cells did not kill the mice. Now that makes sense, right? Because if you kill the bacteria, how's it supposed to be dangerous? Again, that makes complete sense. But it's this next step that was really interesting. So he took heat-killed cells, right, the dangerous heat-killed cells, and mixed them with living, non-dangerous cells. So both of those treatments individually allowed the mouse to survive. But when he did this, it killed the mouse again. And then he extracted a tissue sample from the dead mice, and he found that the dangerous one had appeared again. So what Griffith concluded from this experiment was that that dangerousness was passed from that dead, dangerous one, which didn't kill the mouse, to the non-dangerous one. Some trait was passed, and he didn't know what did the passing at that time. Right? So there's some chemical component that is passing from the dead cells to the living cells. Now remember, they didn't know this, but at the time they didn't know this, but bacteria can pick up DNA from the environment. That's exactly what happened. This non-dangerous bacteria picked up the dead dangerous DNA And now it started making the proteins that made it dangerous, which is why it transformed into the dangerous type. But he didn't know what component, DNA, protein, RNA, because those are the three candidates that they thought were the genetic material. And most leading scientists at the time thought it was protein. This makes sense because proteins do, like everything, they're enzymes, they're transport proteins. We've talked about all these different things that proteins can do. So the leading candidate was some protein that passed it, right? DNA and RNA, they had no idea what their real purpose was at the time. So they were pretty sure that protein was going to be the thing that was the genetic material. Pictures of the different things. Okay. So 1944, it's a long time later. This is almost 20 years later, not quite, that they continued Griffith's experiment. And they were like, you know what we're going to do? We're going to do a similar experiment, but this time we're going to destroy individual components of the strains to see if that dangerousness is still passed. So they wanted to destroy DNA in one treatment group. They wanted to destroy the RNA in another and destroy the proteins in another. So they created three different treatments. So you can say they put the heat-killed S cells in here. They removed the lipids and carbohydrates, and then they added proteinases, it says, or proteases is what we usually call them. So that killed and destroyed all of the proteins. Okay, so there's no proteins there. In this one, they added ribonucleases, which destroyed the RNA. And then in this one, they added deoxyribonucleases, which destroyed the DNA. And then after they destroyed those individual components so they didn't exist within each test tube, they then added those safe live cells, those non-dangerous live cells. And in both the cases, when the protein and the RNA were not present, the dangerous cells still appeared. It was only when they destroyed the DNA that they did not become dangerous. So what did they conclude from this? They concluded that the DNA was the thing that was necessary to be passed from the dangerous heat-killed cells to the non-dangerous cells so that they could develop dangerousness. This was a very important study. They found out, because of this, that DNA is probably the thing that is the secret to how we pass information from one generation to generation. It's what gives us our traits, right? So then, in a separate experiment, not about 8 years later, Alfred Hershey and Martha Chase, or the Hershey and Chase experiment, what they did, and you've actually seen one of your tests had a question like this on it related to this, but you didn't have to know about this at the time. What they did was they radioactively labeled a phage with either radioactive sulfur or radioactive phosphorus. Now, if you remember the rules, right, of identifying macromolecules, if we wanted to label protein, we radioactively label sulfur because that's the unique aspect of protein, right, CHONS. If we want to radioactively label nucleic acids, we would want to radioactively label phosphorus because of CHOMP, right? That's the unique thing in nucleic acids. So, this is showing you what a bacteriophage actually looks like. It's a type of virus that infects only bacteria. It has DNA on the inside, nucleic acids, and then on the outside is a protein coat. They did not know this. They just know it was an infectious thing. That's all they knew. So, what did they do with their experiment? It seemed this is literally like the blender that Miss Bebeck has in her room. They took and they incubated phages in radioactive sulfur in this flask and radioactive phosphorus in this flask. Now notice what's radioactively tagged. They tagged it in pink. When the sulfur was tagged, the outside of the phage was the thing that was radioactively labeled. Again, they didn't know that, but now we know that's how this works, right? And here the DNA was radioactively labeled, so the blue on the inside. So then they mixed the bacteria together with the phages in a blender, and they then took that, after they gave them time to infect the bacteria, and then they separate it in a centrifuge, which spins it really quickly and separates it based on density. So the pellet at the bottom was where the bacteria are, and then the phage stayed up here. Notice the difference of what happened in batch 1 versus batch 2. In batch 1, the radioactivity stayed in the top, right here, where the virus should be. So that means the virus did not pass the protein. But in the second sample, the pellet ended up with the radioactivity. That's where the bacteria are. So what they determined from this was that only when they radioactively labeled the phosphorus did that radioactivity get passed to the bacteria. And remember, phosphorus is synonymous with DNA, with nucleic acids. So this caused the bacteria to become radioactive, and thus we learned that, again, DNA was the thing that was passed, in this case, from the phage to the bacteria. So 2 separate experiments that showed that DNA was the thing that passed the trait on, or the thing that was passed. And here's showing you how it actually works. It injects its DNA into the bacteria, like that. And you can see here, we have these bacteria, these are actual electron microscopy images of the phage doing its job and injecting the DNA. So again, DNA is the genetic material. Once this experiment concluded this, and we also had the Avery, MacLeod, and McCarty experiment conclude the same thing. It was a mad race to determine the structure of DNA, right? So they want to know, like, if DNA is that important, if it's the thing that passes the traits, what is its structure? How does it work? Because if we can figure this out, we'll figure out the secret of life. So Shargov, he was a Ukrainian scientist that actually moved to the United States during the Cold War, right after World War II. And he actually did a separate study, and this is important for the discovery of DNA. He didn't really care, like, he started studying and learning about nucleic acids, but he drew the conclusion through several studies that the amount of thiamine always equaled the amount of adenine. So there's always equal amounts of it. And the amount of guanine was always equal to the amount of cytosine. He never drew any conclusion other than that, right? So, but whenever you had A and T and G and C, the amount didn't always equal each other. So what did he conclude from this? Well, if you remember back from freshman year, when you learned about DNA, A pairs with T in DNA, and G pairs with C. So they're always going to be in equal amounts with each other. A with T, G with C, right? So that he kind of came up with the base pairing rules, but he didn't know that that was the case. But his discovery was still important for determining that. So if you can see here, these are called purines. The A's are purines. They have 2 rings, and pyrimidines have 1 ring, the T's and C's. And notice, the reason that makes it symmetric, when A pairs with T and G pairs with C, it kind of creates a symmetric molecule always. You may ask, well, why doesn't A pair with C? It has to do with hydrogen bonding. We'll talk about that more when we get to the structure of DNA specifically. So then these are the 3 famous people, right? I think almost everyone has heard of Watson, Crick, and Franklin. So Watson and Crick, so first off, Watson and Crick are credited with discovering the structure of DNA. But without Rosalind Franklin, they would never have discovered that structure. She was one of the greatest X-ray crystallographers that was alive at the time. And what you do with X-ray crystallography, show you the picture that she created, you basically beam you beam X-rays through a crystal of DNA that you make. So you make a DNA crystal or any protein molecule, and you beam X-rays through it, and then it creates a scattering pattern on X-ray film. And they use really complicated math. I tried looking this up before. It's like way too complicated for me to even spend time on. But basically they use complicated math to determine the specific molecular structure of of any molecule you do this with. So this is called this is the image, and this is the famous photo that Rosalind Franklin created. But without this photo, Watson and Crick probably never would have came up with the structure. So these are two. James Watson was from America. He came over to Great Britain, and he met up with Francis Crick, and he said, like, and these guys were both kind of nobodies. I don't want to say nobodies, but they weren't like heavy hitters in the field at the time or anything. And he went to Francis Crick, and he's like, we're going to discover the structure of DNA. Like, it's the future. It's one of the most important. It's going to be the most important groundbreaking discovery. Let's figure it out. So they tried to build model after model to figure this out, and they couldn't figure it out. So James Watson decided to go up towards, I think it was King's. I think they were at Cambridge, and I think Rosalind Franklin worked at King's College. It could have been flipped. I can't remember offhand. But James Watson went and listened to Rosalind Franklin do his talk on this photo, and he forgot to bring a pad of paper to jot anything down. So he tried to remember stuff like in his head, which is ridiculous. So he comes back and he tells Crick, and they design a new model. And they're all excited. They think it's the right model. And they call Rosalind Franklin to come look at their model. Now, mind you, Rosalind Franklin was a fairly cold person. And one of the reasons for that is she was a woman in a field where she was constantly ridiculed for being a woman. So she had to be very strong and very cold in order for her not to get stepped on, essentially. Because that's exactly what Watson and Crick are going to end up doing to her. So she goes and she says, she looks at their structure and she's like, this is absolutely wrong. And it's because Watson actually got something wrong with what she said about the numbers of waters. It's complicated, but that were found in the structure. So she's like, you're wrong and you're stupid. Essentially, that's what she said. And she left. And Watson and Crick were kind of upset about that. So what Watson did was he kind of called up Franklin's boss, Maurice Wilkins, and he said, well, I'd like to take a tour. I really can I get this photograph, or can I get this photograph? And Franklin refused to give it to them. So Watson actually went for a tour, and he snuck into her office and stole the photo. And that's how they ended up inevitably coming up with the proper structure of it. So it's this kind of really scandalous story. There's a whole movie about it that stars what's his name? Jeff Goldblum as James Watson. And if you know anything about James Watson, he is a horrible person now. Even he's still alive, which is kind of crazy. Everyone else is dead, including Rosalind Franklin. And Rosalind Franklin died very young. She actually died of cancer from X-ray exposure, because they didn't know that X-rays destroyed DNA, because they didn't know DNA was, or what the structure of DNA was, or how it even worked. So it's this whole really, really interesting, complicated kind of scandalous story in science, and it's a story about one of the most important discoveries in the history of the life sciences. And this is the title of the paper. You can see it was published in 1953, so not that long ago. Most of us probably have grandparents that are alive during when this happened, or even, because some of them could have even been adult, like my grandparents were adults at the time. So I love the last phrase of this, or I think it's the beginning. We wish to suggest a structure for the salt of deoxyribose nucleic acid, DNA. This structure has novel features which are of considerable biological interest. Like, what an understatement, right? It's one of the most, this molecule basically frames all of biology now, which is really interesting. And also, it is only one page long. This is the front and back of a piece of paper. This paper, it's so interesting how like short it was, but all they did was lay out their logic on how they picked out the structure, which their structure ended up being, right, the double helix molecule. And it's a beautiful looking molecule. Look at that thing. It's amazing that it even exists, I think, but it is. It's completely stable. It's a molecule that makes sense based on everything we understand about organic chemistry. And we've seen it now. We've seen the molecular structure of it using electron microscopes, and it actually looks just like that. So this is the molecule that holds the recipes on how we build proteins, how we make all of these things that allow us to live, all the different parts of the mitochondria that did cellular respiration, right? Those different protein complexes. That was all coded for by this molecule. And all of the light harvesting complexes that are found in the chloroplast are driven essentially to creation by this molecule. Okay. And if you notice, the DNA itself is harbored in these things called chromosomes, which are packaged up kind of like really, they're like little hard drives of DNA that you have within your cell or your cell's nucleus. So in 1962, they were awarded the Nobel Prize. As I mentioned, one of my favorite, I usually say in class, if I'm teaching in class, oh, look, notice a bunch of men getting their Nobel Prizes. You can see Wilkins on the end, and you can see Crick here, and you can see Watson here. But notice you don't see Rosalind Franklin. And Rosalind Franklin never got a Nobel Prize for this. And a lot of people were like, oh, it's because she was a woman. Technically, in this case, it wasn't, though I understand that sentiment. But so the rule for all Nobel Prizes is you cannot be awarded a Nobel Prize posthumously. And she died, as I mentioned, of cancer. She would have won the Nobel Prize, or would have been part of it, similar to how Marie Curie won a Nobel Prize before this for discoveries in chemistry. So yeah, just a crazy, wild, kind of sad story. So we kind of as biology teachers, I think we have this really important job of telling the story, because she would have almost been forgotten to history if we in high school didn't tell everyone how she was kind of screwed over by these two guys. So, and as I mentioned, James Watson, this guy's still alive, not a great person. Okay, so just real quick, DNA, what's it made of? DNA is made up of really three main parts, right? Sugars, phosphates, and bases. And we repeat those over and over again. The phosphate group right here, the sugar right here, and then the base, which is either A, T, G, or C, right? Adenine, guanine, cytosine, thymine. So the four bases are, as I mentioned, adenine, thymine, guanine, and cytosine. And they have the exact same phosphate and sugar, it's just the base that would be different in them. So here's showing you each one. There's adenine and thymine, guanine and cytosine. Notice, as I mentioned, adenine is what's called a purine, and guanine is a purine that's because it has two rings, two fused rings. Purines have two fused rings, and then thymine and cytosine have one fused ring, or it's not even a fused ring; it's just one ring, right? Yeah, that's all I have to say about that. And those are called, as I mentioned, pyrimidines. So purines and pyrimidines. Nucleotides will bond together to form a chain, and that chain is called a nucleic acid. So deoxyribonucleic acid, deoxyribose is the sugar that is found in it. And remember, these deoxyribonucleotides are just sitting in the cytoplasm. The most famous one that's sitting there all the time is ATP, adenosine triphosphate. That's a nucleotide. There's also thymine triphosphate, guanosine triphosphate, and cytosine triphosphate. So they have three phosphates, a ribose sugar, and a base. Sound familiar, right? ATP would be the famous one. But we've also talked about GTP, but there's also CTP and TTP around, just in less quantities because they're not so widely used like ATP is. So here's showing you that double helix. It's showing you the hydrogen bonding that occurs between the bases. A and T form 2 hydrogen bonds, and G and C form 3. And that's an important thing to know, actually, because sometimes they ask you questions about that. So, like, if I wanted to, one of the things we can do is we can actually denature DNA by heating it up. And the question would be like, is it easier to denature DNA that has lots of G's and C's or lots of A's and T's? it's much easier to denature A's and T's, so it would take a less or lower temperature to do so because there's fewer hydrogen bonds holding it together, where G's and C's are held together a little bit more tightly with those hydrogen bonds. And those weak bonds down the middle are really important for how it's going to replicate eventually, because remember, all of this backbone is called the sugar phosphate backbone. These are all covalent bonds, which are really tough. They don't break unless they're broken apart by usually an enzyme, or they're burned or combusted. This can be kind of torn apart a little bit easier because it's just an intermolecular force. So the hydrogen bonds hold the two chains together. Okay. So remember the base pairing rules. This is very simple. This should be a review. What is the complementary base sequence here? So it should be, right? Remember, A pairs with T, G pairs with C. So on the other side, it would be T, A, A, C, G, 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 A, T, T. Okay? So where is DNA located in the cell, and what does it actually look like? So DNA is located, or it's contained within the nucleus of the cell, as you can see. When the cell is not preparing to divide, the DNA is just in loose strands called chromatin, okay, which are basically just wrapped around these little proteins called histone proteins, and those histone proteins can unwind DNA so it can be read and turned into protein, or it can wind up and kind of turn those genes off. It's all over the place. And if you notice, it's both red and blue here. That is trying to represent the fact that you have two sets of every single gene. One set you receive from mom and one set you receive from dad. Okay, so you actually have two of every gene. Now those genes don't always have the same versions of them, as we'll talk about when we get into Mendelian genetics, but they're the same gene. They code for the same protein or protein type. Only during cell division does DNA condense to form those chromosome structures that you can see here. And we're going to talk about that more when we get into mitosis. So you can see there's the double helix structure, there's the chromatin, which is wrapped around these histone proteins, and then it gets wrapped and coiled over and over again until finally you get your chromosome structure to really condense it into a little packet that's really dense with genetic material. Sure, let's watch the structure of DNA video. Let's see if it works, shall we? So here's again showing you there's DNA, there's the histone proteins that are important for it to wrap around, and then you can see here, you get these fibers condensing and condensing until finally you get your classic chromosome structure, and this is what it looks like under the microscope. This is going to kind of look, it's not the highest quality video.
6 feet of this long molecule fits into the microscopic nucleus of every cell.
So that's the DNA.
The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again.
And you can see it condensing and condensing. And condensing some more. So loop wrapped upon wrap upon wrap. Think of a chromosome almost like a giant spool of thread.
Leading finally to the familiar shapes known as chromosomes,
And they're showing
which can
a real
be seen
cell
in the
under
nucleus of
microscope.
dividing cells.
This is going to divide.
Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
There it goes. It's dividing, splitting each copy, and then it's going to pinch, forming two new cells. Super cool. All right. So the last thing you're going to do in your notes is I want you to draw the structure of DNA, including base pairing. So you can pause this video to kind of copy what this image looks like. And that's all I have for you. So if you have any questions, ask in class. Ask in class, or come see me during office hours or during lunch. Thanks, everybody.